Okay, welcome everyone to our coffee seminar. Today, I think we should be live now and just wait maybe another minute or two. For the others now, it's lunchtime. It's always tricky, With already 14 online, perfect, 15. So wait another minute before, yeah, we start with the seminar. I think we starting now, just keeping time in mind, and think, numbers are growing online. So anyway, perfect. Joel, maybe you can show the next slide. So again, thanks very much everyone for tuning in today for the seminar. I know it's always really busy times and over lunch and you want to have your lunch, but it's really great, tuning in, coming, attending. So today's seminar is so Joel Johnson, a PhD candidate from CNAS, will present his work about native citrus and the nutritional value and all around. Joel, please, the next one. So Joel is in charge with the slides. So maybe most of you know me. So that's a really, it's really a really old photo. Sorry, I have to replace this. Yeah, I'm academic working at the Center for Nutrition and Food Sciences with Heather and Yasmina. And what I said, I think most of you maybe already met me at one of the seminars or other coffee events. And Joel, please, the next one. Before we start with the seminar, as a tradition, I want to do the acknowledgement of country, and the University of Queensland acknowledges the traditional owners and the custodianship of the lands on which we meet or online meet today. We pay our respects to the ancestors and the descendants who continue cultural and spiritual connections to country, and we recognize their valuable contributions to Australian and global society. Joel, the next one, please. I think you notice when you are a regular uh, participant or attendee of the seminar, so a little bit housekeeping. Joel will then give a presentation around 12:45, 12:50. Then we have a Q&A session around 15 minutes. And please, that's important. Put all your questions you have in the Q&A tab, not the chat. Q&A. And then at the end, you can do this throughout the webinar, and then at the end we have a Q&A session, and I will go through the questions and will read them, and everyone can hear them, and also Joel, and then Joel try to answer the questions. And if there's something super urgent or you want to talk further, there's no problem to contact Joel after the seminar, for some updates. And yeah, I think that's all. And just please keep this in mind, Q&A tab. So Joel, what I already mentioned, maybe not sure, maybe most of you or some of you already met Joel. He's usually working or based at Cooper's Plains, but also doing a lot of work at the moment at Long Pocket, the Alcon building. Joel is a final year or year 3 PhD student at the Center for Nutrition and Food Sciences. He's actually from Rockhampton, so a Queenslander, and it's going very soon. Just told me on Saturday. I thought it's in 2 weeks, but on Saturday to visiting TU Munich for a scientific stay. So he got a scholarship and learning new techniques and analytical techniques there at TUM. And he's very active in publishing. It's already more than 100 papers published, and it's also ranked in the top 2 scientists globally. And yeah, he's working very, at the moment, super intensive on native Australian native citrus, and will present his current or results so far in the seminar today. So Joel, I think the next one, it's all you. Because I can't see the slides, just Joel is in charge. Yes. So yeah, Joel, it's all yours, and please, enjoy the presentation. Thanks very much, guys.
All right. Hopefully you can hear me all good. Thanks for the introduction, Michael, and thanks for the acknowledgement of country and the traditional owners as well. So today I'm presenting on my PhD project, the composition of Indigenous Australian citrus fruit, particularly on their polyphenols, volatile profiles, and other aspects. We can get these slides to work. So to start us off, the question is why am I looking at citrus of all the types of food we could look at? Well, citrus is the most widely grown tree fruit crop in the entire world. And it's also been classified as one of 35 food crops which are essential for conserving and developing our diversity for a balanced and sustainable diet. Globally, the citrus industry is worth over 89 billion US dollars, which is around 130 billion Australian dollars. And this corresponds to more than 140 million tons of citrus fruit being produced every single year. So it's a really massive industry. And the export market is, again, around 15 billion US dollars. So it's really significant from an economic perspective. And it's important in other ways than that, because without citrus, what would James Bond have in his martini? Without citrus, how would we come up with corny phrases like, if life gives you lemons, make lemonade? And without citrus, what would Paddington Bear eat? So citrus is a really important part of not just our economy, but of our diets, our nutritional intake, and also our cultural icons. And it's also quite a big industry in Australia. We can measure this using a classic indicator of the importance of something in Australia, which is the big thing. So in Australia, we have the big orange in Berry in South Australia. We've also got a big orange here in Gayndah in Queensland. And then we've got a third big orange in Harvey, Western Australia. And on top of that, we have a big mandarin just a little bit down the road from Gayndah in Mundubbera. So by all metrics, citrus is a massive industry globally and also in Australia. So in Australia, we produce around, it's just past 1 billion in value per year. That number on the screen, I think, is from 2024, but in 2025, the industry cracked $1 billion in value, around 760,000 tons every year. And so I've just put on the screen for you these fruit, and the size of the fruit corresponds to the proportion of production. So biggest type of citrus that we produce at the moment is oranges, followed by mandarins, around a third of the amount of oranges. And there's also a decent production of lemons and a very small amount of grapefruit, which is produced. Most of this takes place in southeast Queensland, as well as sort of the Riverina area in New South Wales and Victoria, and the Riverland in South Australia, as you can see on that map there, where the green parts are areas of citrus production. But there are challenges to our citrus industry. To quote Agent Smith, well, not really, but to pseudo quote him, we all know that bacteria are a disease, but more specifically for our purposes, Huanglongbing is a type of disease, a type of bacterial disease, and it's emerged from China and it's attacking citrus industries across the world. So you can see here, this bacteria gets into the leaves and turns them all yellow, which means that the citrus plants can't photosynthesize, and it also really damages the fruit. And so this disease, Huanglongbing, has devastated citrus industries in China and Asia. It's gotten into the US recently, parts like Florida and California have lost 90% of their citrus production to this disease, Huanglongbing. So their current production levels are 10% of what they used to be a few years ago. Now, Huanglongbing hasn't reached Australia yet, but it's in Papua New Guinea, just across the Gulf. And it's only a matter of time, we expect, until it gets here. And we're not prepared because no existing varieties of citrus which we grow are resistant to this disease. And there's no real effective treatment for it. As well as this major threat of Huanglongbing, there's also challenges from fungal diseases, from changing climatic conditions, because citrus is very sensitive to temperature, drought, and saline soils in particular. And then there's economic challenges because citrus tends to be a fairly low margin crop, fairly labor intensive, and there's a lot of cheap international competitors who from Asia and the US who can bring their citrus fruit into Australia and compete with our growers at quite low costs. So how do we solve a problem like Huanglongbing, climate change, and low profit? So there's a bunch of organizations looking at that, including Citrus Australia, different state governments, Hort Innovation, and a bunch of universities. So just like in Star Wars, the solution to a lot of these challenges is the resistance, because recent research has found that native Australian citrus have genes which make them resistant to this disease, Huanglongbing. They're also adapted to Australian environmental conditions and resistant to many other diseases like the fungal disease I mentioned. And so a bunch of recent studies have highlighted this, and the global citrus industry is really looking to Australia at these native citrus species to see whether we can increase commercialization of them or maybe use their genetics for breeding into conventional citrus varieties. So just to touch on our native Australian citrus, we have 6 species in Australia, which is the most number of species native to any country in the world. We've got Citrus australasica, the finger lime, which grows around southeast Queensland. Also, Gympie lime, Citrus australis, which has a similar distribution in the subtropical areas of southeast Queensland. Then in far north Queensland, in the rainforest, we have Citrus garraway and Citrus inodora. And they've got you'll see here from the photos that our citrus have very different characteristics to what you might think of like an orange or a mandarin. A bunch of these have like an elongated shape, different colors. So there's a lot of novel aspects of them because these are all species drawn straight from the wild. They haven't gone through the domestication process, selecting for like shape and color. In the Northern Territory, we also have Humpty Doo lime, Citrus gracilis. And then out in Western Queensland, New South Wales, we have Citrus glauca, the desert lime, which is the only xerophyte in the citrus genus, which is a xerophyte being a plant adapted to dry conditions. So our native Australian citrus might be a potential solution to a lot of the problems that the citrus industry is facing globally and especially in Australia, because as I mentioned, they have this resistance or tolerance of common diseases, including Huanglongbing. They're also adapted to Australian conditions. Some of them are tolerant to drought, like the desert lime, also heat, frost, salinity, all those things which tend to plague our conventional citrus production systems. And that means there's a potential to expand our production areas further north and further west compared to where they currently are, which tends to be along inland river systems. And because of their novelty, they also have the prospect of being a higher value crop. So that helps on the economic front. But the main challenge is there's very few scientific studies at all on our native citrus species. So we don't know much about the nutritional quality or their anti-nutrients, if there's any. Like, are they safe to eat from a scientific basis? Do they have any health benefits? Do they have the health benefits of our conventional citrus? Do they have added health benefits on top of that? And does their composition change a lot depending on the geography where we grow them and on the season? And so that's where I step in, looking at this chemical composition aspect. And the indigenous use of these species is there, but it's largely undocumented in the scientific literature. So citrus glauca has been mentioned to be used for food and medicine. Citrus australasica, that's the finger lime, has anecdotal reports of it being used to prevent illness and as an antiseptic. There's mention that Citrus australis, the Gympie lime, was eaten by indigenous tribes, and Citrus gracilis may have possibly been eaten, although other anecdotal reports suggested it tastes a bit like tar. So maybe there was some preparation, or maybe it wasn't actually eaten, and that it just like a that mentioned there is a footnote in a larger article. So possibly the author got that wrong. But this is, as I mentioned, this is where I step in to look at the composition of these native citrus fruit from a scientific perspective, all their nutrients, anti-nutrients, and health benefits. And so my motto for this project is, when life gives you lemons, instead of making lemonade, I'm going to chop them up, analyze their nutritional composition and bioavailability, investigate their health benefits, and discover new compounds. So in a more scientific form, we have our native citrus fruit, which undergo chemical analysis to look at their nutritional quality, investigate them for new compounds, look at their geographic and seasonal variability, also how these nutrients are taken up in the gut, whether they're bioaccessible, and then test their bioactivity, with the ultimate result being to determine the health benefits of these species compared to conventional citrus. So I'm going to run through some of my results so far. So when we're looking at vitamins and some other nutrients, if you look at the vitamin C content in the graph here, you can see all the pink ones are the finger lime, Citrus australasica. This purple one here is Citrus australis, then the light green Citrus glauca. We have Citrus inodora in yellow, Citrus garraway in blue, and then on the far right in the dark green, we have Tahitian lime. So that's the lime you would get if you go to Woolies and pick up a lime from the store there. They're all Tahitian limes. So that's what we use as our benchmark standard citrus to see whether the native Australian limes are comparable or doing worse or better than Tahitian lime in terms of their nutritional composition. So when we look at the vitamin C content in the flesh or the pulp, all of the species except for one had significantly higher vitamin C compared to Tahitian lime. And this was up to 4 times higher in our Citrus inodora and almost 7 times higher in Citrus glauca, that's the desert lime. So if we look at on a fresh weight basis, this best performing species, Citrus glauca, had almost 200 milligrams of vitamin C per 100 grams. And as the RDI set for vitamin C in Australia and New Zealand is 45 milligrams per day. So that means you'd only need to eat 25 grams of this particular native Australian citrus to reach your complete RDI. And that's several times lower than our conventional citrus, as you see here. In terms of, you'd have to eat a lot less of that to reach your RDI. The native citrus also have tend to have a higher dietary fiber content, which is good for improving digestive health and disease prevention. So it's reaching up to 7% dietary fiber in some species, particularly in Citrus garraway and the Citrus australasica, the finger lime. We also looked at their sugar profiles in collaboration with partners from Queensland Health using ion chromatography with pulsed ampermetric detection and confirming the identity of all these sugars using mass spectrometry. So you can see a chromatogram there. And when we compare native citrus to Tahitian lime, the conventional commercial citrus variety, most of them tend to have a lower sugar content, which means they end up tasting a bit more sour. And again, this is to be expected because they're wild germplasms. They haven't gone through selective breeding processes, which tend to increase the sugar contents and make them more palatable. And so some of this might be able to be changed through selective breeding. But in general, except for, there was a few of the finger lime varieties on the left here, which had a higher sugar content. So we have sucrose at the top, fructose in the middle, and then glucose in the bottom graph. And one thing to note is that the native citrus, while they had a lower level of sugar overall, they had particularly low levels of glucose in all these species here. In the finger lime on the left, the glucose level was roughly comparable to Tahitian lime, but in all the other species, there's virtually no glucose in there. And when we look at the glycemic index of our common sugars, glucose has the highest. These are the ones like sucrose and fructose have a lower GI. And so that means, just based on these preliminary results, we might expect that the native citrus will generally have a lower GI compared to conventional citrus because it has lower levels of glucose and high levels comparatively of these other lower GI sugars. The organic acid content is the other half of the equation when we're talking about citrus flavors, because it's really the balance of sugar to acid that determines how sweet or sour it will taste. So most of the native citrus had a comparable level of citric acid, which is the main organic acid in them. It was roughly comparable to our commercial Tahitian lime. However, the desert lime, Citrus glauca, had much lower level of citric acid here, around 3 times lower than conventional lime or other native lime species. And so that means we expect it would have a much sweeter flavor, whereas most of the native citrus tend to be quite acidic and sour. And then we also looked at some of the other vitamins. So vitamin E is a really powerful antioxidant, which has a role of scavenging free radicals in the body. Also helps boost the immune system and prevents plays a role in preventing chronic diseases. When we look at the levels of vitamin E, the compound alpha-tocopherol in the flesh of our native citrus, most of the species had a comparable level to Tahitian lime. There was a few which didn't have any detectable vitamin E, but there was also one standout here, Citrus inodora, one of the species from far north Queensland, had almost 3 times the level of vitamin E compared to our conventional Tahitian lime. And when we looked in the peel of these species, which isn't normally something eaten, we do use a little bit of citrus peel in the zest, but it might have particular significance if we extract the compounds from there and use it as some extract or some value-added product. But when we looked at the peel of the native citrus, there was a massive amount of vitamin E in them, much higher, more than 30 times higher than Tahitian lime in some of the species, particularly in some of these finger lime varieties here. So these could potentially have use in, as I mentioned, skincare products, because another role of vitamin E with its antioxidant properties is in protecting skin, helping healing and preventing aging of the skin. And so there's quite a potential here with this massive amount of vitamin E that these, it could be extracted from these finger lime variety peel and used in commercial products. Moving on to another important vitamin, provitamin A. So mostly beta carotene and other related compounds. So the compounds which give carrot its orange color. So these are, again, very powerful antioxidants which help protect cells from free radicals, antioxidative oxidative damage, and also play a role in supporting the immune function. So most of the citrus didn't have any of this provitamin A in the pulp. However, a few of the native species did, particularly Citrus glauca and 2 varieties of finger lime. These are moderate levels because the RDI for beta carotene is about 11 milligrams per day. And so with our beta carotene content here, you'd have to eat quite a significant amount of these to reach that. So it's more useful perhaps as a supplementary supplementary aspect of the nutritional value. Again, we looked at the peel, the beta carotene in the peel of these species, and pretty much all of the native a native Australian citrus had equal or higher levels of beta carotene compared to Tahitian lime. So most finger lime varieties were around 2 times higher. The Citrus australis was more than 3 times the level of vitamin A compared to Tahitian lime here. So again, the peel is not likely to be eaten by general consumers, but it might be useful for extracts or other cosmetic applications, that type of thing. Now, another important aspect, particularly of citrus, is the polyphenols and flavonoids. And these are a range of compounds which are associated with a bunch of health benefits, including anti-inflammatory effects and improving cardiovascular health, because they have strong antioxidant activity. So we looked at the total antioxidant capacity of our samples. So this is basically how many antioxidants of all types are in the samples. Most of the native citrus actually have a bit of a lower level of antioxidants. But again, the citrus glauca was a standout with more than double the amount of antioxidants compared to Tahitian lime. And so the citrus glauca is one of the species we're quite interested in exploring further. Now, we looked at the specific polyphenols that were present because, well, having a picture of the total amount can be a bit useful. It really depends on the individual compounds that are present because the biological activity changes drastically depending on the specific polyphenols which are present. Because some will have beneficial effects, some will have negligible effects, some might even have negative effects on health. And so that's really a really important thing to look at is the specific polyphenols that are there. And we did this using liquid chromatography mass spectrometry. And so these are just the results I'm presenting from one of the native species, Citrus australis. So we found a bunch of compounds like naringenin, and compared to our Tahitian lime, this was more than 1,000 times higher in the Citrus australis, the native species. And so this one is obviously got much higher levels in our native citrus compared to conventional citrus. And these are a bunch of compounds which are associated with antioxidant activity and improving cardiovascular health. We also had isoramnitin at high levels, gallic acid, another common polyphenol, naringenin, which is the similar compound to naringin, but without these glycosyl groups there. And also eriocitrin, which is a characteristic polyphenol found in citrus. We also looked at a more specific class of hesperetin and its derivatives because these are associated with anti-inflammatory and neuroprotective properties. And there was a bunch of these found in the native citrus, although they tended to be at lower levels compared to conventional citrus. Things like hesperetin, the hesperetin 7-glucoside, hesperidin, and neohesperidin. They're just glycosylated forms of that hesperetin flavonoid. But all these compounds were more abundant in the peel of our native citrus compared to the pulp. So this might mean, again, extracts of the peel might have additional health benefits on top of the higher levels of vitamins like vitamin E and vitamin A, which we mentioned previously. And so that's something to consider is the composition of the peel as a whole. So if we use that for extracts, it might have great activities than considering just the components alone. The final group of the compounds we wanted to focus on was polymethoxyflavones. And so these are a very specific class, specific to citrus, a class of compounds which have a strong association with anti-obesity effects, amongst other beneficial effects. So we found actually the most common polymethoxyflavone called nobelitin in our Citrus australis species using LC-MS spectrometry. And you can see the structure there. So they're called polymethoxyflavones because they have a bunch of the methoxy groups, these OCH3 groups, all attached around this flavone skeleton. And so that's what gives them their potent bioactivity. And so we can see in the mass spectra here the loss of these 15 Dalton groups from some of the methoxy groups, and then also the loss of this ketone group here. So confirming the identity of nobiletin. We also found a few other polymethoxyflavones which were unique to our native citrus. So they weren't found in the commercial Tahitian line, like piloine we have here, and then another glycosylated form of the polymethoxyflavone right here. So these are something that we're looking into further, trying to investigate. So that was the results from just one of the native species. We're looking at all the other ones, and then also potentially looking at trying to quantify these. So see not just what type of polymethoxyflavones are there, but also how much is present. And then the final aspect of the composition, which is really important, again, for citrus, is the volatile composition, because this plays a role in our sensory perception. So when the consumer picks up a fruit, the smell has a really big impact on whether it seems desirable to the consumer. And it also has an influence on the taste, because both the volatile, the odor, and the flavor impact how we perceive the taste of fruit when we're eating it. So when we look at our finger lime, the volatile, so this is a GCMS chromatogram here, the volatile composition is really dominated by this compound called D-limonene monoterpene, which is very common in citrus. But the finger lime volatile profile is really unique in just how much D-limonene is present. So in that term, you can consider a very, very simple volatile profile, but we did find more than 100 minor compounds present. As you can see, some of these like beta-pinene and a bunch of other compounds at low levels. When we look at the genome of finger lime, which has recently been sequenced, it contains a bunch of unique genes for terpenoid backbone and monoterpenoid biosynthesis, which is responsible for all these, as I mentioned, more than 100 minor compounds present. Now, the Gympie lime, Citrus australis, has quite a different composition. It's got alpha-pinene as the dominant compound, much lower levels of D-limonene, and so it's quite a drastic shift in the volatile profile compared to the finger lime. So alpha-pinene has more of a woody sort of piney scent, and it was mostly monoterpenoids in this species here. And that actually makes sense when we look at the genome of this species, because it contains 37 genes for monoterpenoid synthesis compared to only 24 for sesquiterpenoids. And so we're getting sort of a match between what we predict using the genes that are present in the genome of the species and then what we actually see in the volatile profile here. And on top of that, it has 10 of these genes are specifically for encoding beta-myrcene and beta-ocimene synthase. And so we see a fairly high level of beta-myrcene coming through. Now, in Citrus garraway, the Mount White lime, it was quite almost flipped compared to the Gympie lime, because we've got a bunch more sesquiterpenoids, these larger, heavier compounds such as copperoine, caryophyllene, delta-catenine coming through. There's a bit of D-limonene, but again, much less compared to our finger lime, the very first one that we looked at. When we look at the genome of Mount White lime, you can see it's enriched for the backbone biosynthesis of terpenoids, and so that means that it's likely to make these larger backbones of the sesquiterpenoid compounds, which again we see coming through in the volatile profile here. Now, the desert lime, Citrus glauca, has a bit of everything. So it's got a few of those compounds like alpha-pinene and hexanal, a bit of D-limonene, again less compared to finger lime. Finger lime is really unique in having just D-limonene as pretty much the dominant compound. But Our desert lime here also has a bunch of the sesquiterpenoids, as well as the monoterpenoids. And then finally, Russell River lime, the Citrus inodora, did have D-limonene as the most abundant compound, but it also had moderately high levels of these other compounds like beta-pinene and gamma-terpenine. So mostly monoterpenoids, a few sesquiterpenoids here. And it tended to have fewer peaks overall. So out of all the 5 native species we investigated, the Citrus inodora had the fewest number of volatiles identified. And that kind of matches up with earlier descriptions of the species, because the scientific name Citrus inodora comes from the Latin inodora, which means no odor. And that was the observation of the early botanists who encountered this species and observed that the flowers and fruit tended to have less odor compared to other citrus species. And so that, if you like, qualitative anecdotal observation matches with what we find when we actually look at the volatile profiles here, where there's a lot fewer volatile compounds compared to other native species. So if we just overlay the chromatograms here, you can see there's a bunch, there's quite a wide variation in terms of their volatile profiles. So a few of them being dominated by sesquiterpenoids, particularly Citrus glauca, Citrus caraway, and then other species being dominated with fewer monoterpenoids, but tended to be at higher concentrations. So things like the finger lime, Citrus australasica, also Citrus australis. And then this is just a quick summary of the number of volatiles found, because we identified hundreds of volatile compounds across the species. Most of our native species we found, between so 51 was the lowest found in Citrus inodora, all the way up to 149 different volatile compounds in Citrus caraway. And so there's hundreds of these volatile compounds here at different levels. And so we're still in the process of identifying some of these and then comparing like which volatiles are unique to our native species compared to which are found across most citrus species. Well, there's definitely some unique chemistry going on and more things to explore. Yeah. As I mentioned, D-limonene tended to be a major volatile. So it was the highest level in most species, which is fairly common for citrus species, particularly in Citrus australasica. It was also the highest, the most abundant compound in Citrus caraway, Citrus inodora, and the same in our conventional Tahitian lime. And in those species where it wasn't the most abundant, then alpha-pinene, that more woody, piney scent, tended to be dominant in Citrus australis and Citrus glauca. Now, we haven't done any consumer studies, but there might be something worthwhile considering down the track towards commercialization, whether these two species have the same attractive citrusy scent as the other ones, or whether they might have a different scent which might be less attractive to the consumer. That's just conjecture, obviously, at the moment. And this is just in graphical form, just showing that 2 of our native citrus species were very high in this D-limonene. So that was Citrus inodora and Citrus australasica, kind of at higher levels, similar levels compared to conventional citrus. So things like Tahitian lime. We also did testing on lemons and oranges. They all tended to have high levels of D-limonene, whereas the other native citrus, while it was present, tended to have quite low levels of D-limonene. And then finally, alpha-pinene, as I mentioned, this monoterpene with more of a piney, woody odor, was kind of like a marker almost for several native species because it was found in such high levels. And sorry, just go back. Particularly Citrus australis had quite high levels of this compound. Almost 60% of all the volatiles present comprised alpha-pinene, and also Citrus glauca had a decent amount of alpha-pinene as well. So I think we're coming up to around the end of my time. So I'll just wrap up with a few points. So when we're looking at the composition of our native species compared to our conventional Tahitian lime across all the different testing that I've done so far, just a few pull-out points. Some of the species had 4 to 6 times higher vitamin C content, which is very promising in terms of their nutritional composition and health benefits, and also potentially useful for marketing these species as we look toward broader commercialization of them. They also had a higher dietary fiber, which is another important aspect of the nutritional composition of citrus. They tended to have a lower sugar content, except in a few finger lime varieties, but fairly similar levels of organic acids, which means because they've got similar levels of organic acids, but a lower sugar content, they tend to taste a lot more sour than conventional citrus. The exception to this was citrus glauca, which had much lower organic acids. So that might be a more sweeter um, type of our native citrus. They also had really high levels of vitamin E in the peel, up to 33 times higher than Tahitian lime in some species. Their polyphenol profiles were characteristic of other citrus, including things like hesperetin, arginine, isorhamnetin. And we also found several unique polymethoxyflavones in one of the native species, which we've tentatively identified. And these are, again, the polymethoxyflavones are the class of compounds associated with specific health benefits, particularly anti-obesity activity. And then finally, their volatile profiles were quite distinct, with D-limonene tending to be the dominant compound. And so there's another novel aspect there, in addition to their unique shape and colors. Some of them might have unique or distinct aromas. So I'd just like to finish by acknowledging all of our suppliers, including Herbalistics, Professor Robert Henry and Yupali from UQ, Rocanda Regional Council, Green Valley Finger Lime, Salty Lime Co., and Australian Desert Lime all supplying samples. And then also from UQ, Toa Nguyen and Ming Li for helping with the analytical methods. So with that, you're probably thinking, aren't you glad this is the end of my presentation? And I'll wrap there and happy to take any questions.
Thanks very much, Joel, for the presentation and the slides. And so we have at the moment 2 open questions, and one it's kind of a comment, and it's more like comment on your stuff, Joel, and I think it's from a grower breeder. But first, it's from Carlos, then from Carrie. So first question from Carlos: any reason some parameters, including dietary fiber, sugar, and sugar content, was not analyzed in citrus glauca, Joel? Maybe didn't show. I can't remember.
Yes. So the citrus glauca was the last of. So I had to collect all these species at different times of year, and actually across because they all fruit and ripen at different times of year. And so citrus glauca was the very last of the species I've collected. And so we have done the dietary fiber and sugar contents on it, but I haven't had the time to update the graphs, if that makes sense. So it was just because we did all the testing on the other species first, and then I got the citrus glauca, and I haven't got to adding that to the graphs yet.
Mm-hmm. Okay. So hope, Carlos, that's for the moment okay. And when you look, if you want to further updates what I said before, and Joel had his last slide, so and he's in the UQ system, Quavi system, so just send the email and I think get directly in contact. So maybe that's the easiest way.
Happy to share.
Yeah. So then second
updated
question
data as well.
is from Carrie, our Carrie, and how are representatives from different additional lands involved in decisions about sourcing, cultivating, and analysis of native lime species?
In our study, we've sourced the samples from commercial suppliers. So these species are already so there are some commercial farms of finger limes around, not many compared to other commercial citrus like oranges, mandarins. There are a few small farms of commercial that commercially grow finger limes, and so we've sourced our samples from those, so through more commercial channels, and some of the other species from plant nurseries. And so we haven't gotten these samples from traditional lands, if you like. But it is something which is worth noting is that they're already grown, some like the finger lime grown around the world. And so because this germplasm has sort of been taken to other countries, and so the biggest finger lime plantation in the world is actually in I think it's Guatemala or somewhere. And so the representatives of like the finger lime industry here are quite interested in getting more research done on them in Australia, so that it doesn't end up like, say, the macadamia nut, so that we can show the benefits of Australian grown citrus, and sort of center them as being from Australia. Because as you know, the macadamia nut was taken to Hawaii, and now in America they know it as a Hawaii nut because they dominated the production and then also the research into it. So I don't know if that really answers the question. The short answer is that we got all our samples through more commercial channels for this
Maybe
study.
maybe Carrie, or you can maybe also after the seminar directly talk, or if there are any no, that's always important question, and they don't any benefit in everything. Adam, there's another question now from Adam. So please feel free, guys. If you have questions, you can still type them in the Q&A. That's fine. So there's another question from Adam. You used lime as a control in the vitamin A test. So it should be provitamin A. So vitamin A is usually not in plants. Limes and sorry. In the vitamin A tests, limes are famously green. Do the native limes still have more vitamin A, beta-carotene than oranges or lemons?
Yeah, so I've only showed the Tahitian lime in the graphs just to try and keep things fairly simple, like a little complicated as it is, but just as the one control group. We also did test oranges and lemons for all of these analytes. And yes, I believe the native citrus. So in orange and lemon, it was a little higher for the provitamin A compared to Tahitian lime, but the native species, so the finger limes, still had much higher levels of these provitamin A compared to even oranges and lemons. And you can kind of see that in some of the finger lime varieties are a lot more red or yellow or orange, and so you can see that coloration of the provitamin A there as well.
Next question from David. So that's a good one. So far all questions are really important, good ones. So don't get me wrong. So great work. Why were just pulp studied, not juice, to make the whole picture? That's an important one. As juice is one of the, let's say, most popular, always drinks from citrus fruits, and yeah.
Yeah. So for the pretty much all of the native Australian citrus species tend to be a little bit drier compared to other citrus. And so things like finger lime, if you squeeze a finger lime, you're going to get hardly any juice out of it. And so there might be some use if you have a really large quantity of fruit and try and juice it, you might get a small amount of juice out of it. But I don't think that will be the main use of these native species going forward, just because they tend to have less juice than other citrus, which we're used to. So for the pulp sample, what we did is we took the entire, basically like the inside edible portion of the fruit, is what that corresponds to. When they come in, I separate it. So I took all the edible portion. So, and that included the any juice and also all of the flesh. So most of the native species, the juice is contained in little vesicles. So kind of like when you have an orange, you'll see there's little portions that have all the juice in there. And so I've took all that without breaking them, and then have dried that and tested it. And so all of the parameters is basically for the pulp is basically the whole edible portion, which includes like the I guess like flesh and the juice.
I hope, David, it's fine. But that's a good question because usually, what you said also from the limes, I mean, Joel, I think your flag is that you didn't, or what is not a focus on, or you can't do everything, in your PhD now, sensory and consumer acceptance. But I think this is also now, when it comes to product or product development or anything, or just what you're saying, like choosing, a really,
Yeah,
really significant question. If it's from the sensory, then it's maybe more, will be more used for blends, to blend with others, but maybe not as a, only, you know, fruit for only the, like, I don't know, citrus glauca, for example, for juice, what you said. It's because this is at the end, I think, for consumer acceptance and any market success, still the most important one. If it's even, we have lots of health bioactive compounds and very healthy composition, but if it doesn't taste or is what you said, astringent or, sour, then it's very, very difficult to as a poor product, maybe then as a blend, with others. I think that could be but that's something maybe beyond uh, just thinking. And what you said, some are relatively dry, then it's really hard to get any juice out of it.
Yeah, we did originally have a plan to juice some of them and study the juice, but
Would
what
be
I
good,
found
yeah.
when we came to do that in the lab
Mm,
is we'd get like less than 1 ml of juice from the fruit just because it's got less,
mm,
and it's also in those vesicles, which are really hard to break. And so
yes.
yeah, it's really unfeasible to study the juice.
Right. And I think from an economic perspective,
Yeah.
definitely maybe won't be a feasible or, product. Look, another question from up dot one from, I don't know what's, sorry, your real name, sorry. Um, was there differences in chemical composition across samples harvested at different times and locations? I mean, that's also really important. Joel,
Yes, obviously something that I didn't present here. I have collected some samples across different seasons, so from the same location across 2 consecutive seasons. And there does seem to be some minor differences in the analytes that we tested, things like vitamin C. I think from memory, it varied by about 20% um, up and down from one season to the other. But we haven't been able to do a full um, comparison of like across a bunch of seasons or across a bunch of different locations because a lot of these species are quite hard to source. Some of them it's hard enough to get hold of fruit from just one location. But that's definitely something probably for future work to build on. A lot of my PhD is doing sort of getting all the baseline data for some of these species which haven't been investigated before. So like we don't even know the vitamin C content or vitamin A content or vitamin E content of most of these species. So I'm pretty much the first person to study and hopefully publish that in the near future. And so mine is more doing sort of the baseline data, and then it'll probably be up to future research to do more detailed investigations of how that varies across different locations and different seasons. But from the limited work that I've been able to incorporate into this, it does seem that there's a small to moderate variation across different growing conditions.
hmm No, thanks, Joel. Look, we have two more questions, I guess, and then maybe the last two for just thinking about the time from I and then from Kerry. First, I. So thanks, Joel, for a great presentation. Have you had a chance to look at the, economics data as I did not see you presented in this presentation? It is interesting to know the macro and minor nutrient profiles among species.
Yeah, which data was that? Sorry.
The economics, maybe the minerals, trace elements,
Yeah. Okay. Okay. So,
and no
yeah.
macro and minor or macro
Yes.
and
Yes. So
micronutrients.
yeah. So we have done the yeah macro and minor minerals on some of the species, comparing them to conventional citrus.
It's also
Um,
from I, the question.
yes. Yeah. Yeah. With I. And so hopefully we'll be
Mm.
publishing that in the near future too. Just wasn't able to incorporate it into this presentation without it going massively over time. Because there have been a bunch of additional tests like the minerals and other things which we've done. From memory, the mineral content was fairly similar to other citrus, which is probably good because we don't want anything too wacky going on. And the mineral composition also depends a lot on the soil. So it's probably more to do with the growing environment of the citrus rather than their genetics specifically. But it tended to be fairly similar. We didn't see any elevated levels of heavy metals, which is really good. We don't want to see that. And it had very low levels of things like sodium in there, which again is a good from a nutritional perspective because that can, produce elevated blood pressure. And so there was definitely some interesting things, but nothing too unexpected in the macro mineral, micro mineral composition.
Thanks, Joe. So last question from Kerry. I noticed that your references include historical articles, such as those from the 1800s. Could you share more about your methodological process specifically? Did you incorporate oral knowledge from indigenous traditional owners or use workshop-based engagement alongside literature review? How were those, sorry, this is a small, how were these different sources of knowledge balanced in your approach?
Yes, so the references I had are all pulled from my literature review my literature review summarizing basically all the scientific literature on the native species. I haven't been able to do, like you're saying, that sort of oral workshop gathering traditional knowledge as part of my project because my background is chemistry and I'm really focused on the composition. But that's definitely something that's really important to do is to gather that traditional knowledge. But that obviously takes a lot of time and resources because of all the ethics around that. So I wasn't able to incorporate that aspect, although it's very interesting and important as part of my PhD. So that's definitely out there for somebody to use. So in terms of the background, I'm mostly interested in previous reports on the composition of these species. I've just been working from the peer-reviewed scientific literature.
Oh, look, I think it's now 12:59, almost in time. And sorry, I apologize for my sometimes I had problems to read the stuff. It's on my small screen. You're next to my main screen. It was really, really small, so a bit difficult sometimes to read. Apologies for that. So thank you so much again, for all of you zooming in and spending your time. I think we have, and Joel, thanks so much for your presentation. And I think we have one more slide. I think it's the last one, Joel before. Yeah. So the next graphic seminar, it's in 2 weeks, in a fortnight. As you know, it's always fortnightly. It's from Palakdeep Kaur from, I don't know which sender. No sender, maybe Hotside. I don't know. Yeah. Anyway, and she's will talk about investigating the genetic variation and mechanisms of cell fertility in macadamia. Sorry, Palakdeep, that I don't know your sender affiliation. Sorry for that, but maybe anyway, you will get an email, the usual invites, and have a look at this. And I think there's another one. Sorry, Joel. I think the ultimate last slide. Joel is gone. No, he's here. Look, and if you wanna be added to the list of the seminar list, the invitee list, so just scan the QR code. That's the easiest way, and then you are on the list. You can also send the email maybe to Megan or have a look, at your list via email. And I would say it's definitely worth, lots of really interesting seminar topics from, the whole range of agriculture, food, nutrition. And yeah, it's always, I think, always a Tuesday highlight, a lunchtime highlight. And yeah, that's, I think, everything for today. And thank you so much for that. And if you have further questions, so please feel free to contact us or Joel directly. Also, Yasmina. Yasmina is also one of the co-supervisors. Maybe Carrie, that's also worth, talk. And yeah, see you then in a fortnight. Thanks very much, guys. See you then. Bye.